and the classification and production baker yeast, and the harvesting of yeast, drying method of yeast, and next one is the drying method of yeast, and these are things are we will discuss today. And this fungi is the how to use for this one production filamentous of fungi, and this one how to use, and that characters are that fungal. And these are things and critical parameters of production of fungi. Next one is the recent market value of the fungi. Um, Recent value of the fungi, and that these are things you'll discuss about to the fungal classification and filamentous, how to source are these one. Now we will discuss about to the overview of the fungi. What are the fungi? See, the fungus, most, that is the useful fungi is there. And now sometimes are the different types. Fungi are industrial important organisms due to a wide range of applications, including in a biotechnology, in the production of the food products, organic acids, pharmaceutical products, polysaccharides, and enzymes in biological control of pest, insects, fungi, and weeds. Ball and this one to identify the explanation for the Ball and Finkelstein in 1992. The fungi are known to produce a variety of secondary metabolites and have been exploited for the production of homologous or heterologous gene products due to the efficient protein creation mechanism. In the these are the explanation. Domes, Knowles, Sundars, and these are scientists. The fungi are eukaryotic and heterotrophism. The fungi cell structure is the some eukaryotic and some are the heterotrophism. These are not to prepare the food material. That's why these are all called the heterotrophism organism, and that are most commonly known as for their ability to grow and digest waste organic matter also. The fungi range from multicellular, the starting from the multicellular to macroscopy, from like that of mushrooms. Mushrooms also one type of fungi. That is you can see up there in the directly eye contact. That is called the macroscopic structure. And macroscopic form like that of mushrooms to unicellular microscopic forms like that of yeast. That is a very minute. Yeast, that is the unicellular organism, fungal organism. The mushroom also, that is the broad structure or macroscopic structure. The yeast is the microscopic structure. Yeast is the microscopic forms like that of unicellular microscope like yeast, with over 1,000 or 1 lakh species classified as the fungi. Fungi can be divided into morphologically three groups. That is called the morphologically three groups. Their morphology. First one is the yeast. Second one is the dimorphic fungi. Third one is mold. Here, these are unicellular organisms with nucleated round fungi that reproduced by the binding. An example is Saccharomyces cerevisiae. Dimorphic fungi, these grow partially as yeast and partially as filamentous fungi, depending on the environmental condition. For example, Histoplasma capsulatum. And this is a yeast. What is the structure of yeast? Yeast is a unicellular organism with nucleated rounded fungi that reproduced by the yeast always reproduced by the budding. Best example is the Saccharomyces cerevisiae. Dimorphic fungi. Dimorphic fungi is there are grow partially as yeast, partially as filamentous fungi. Depending on the depending on the environmental condition, depending on the environmental conditions. For example, Histoplasma capsulatum is the dimorphic. Another one is the mold. These are multicellular organism. Always you can observe to the molds, multicellular organisms all are called the molds. These molds, the structure of mold is the filamentous in nature. Filamentous meaning the plant body or fungal body, filamentous in nature and hyphae also present. When these are in the reproductive time, the hyphae reproduce the spores or through the spore. For example, Penicillium chrysogenus. You know, best example, Penicillium mold, green mold or blue mold, black mold, like this. Different molds or different color molds also present. That's where the total fungi, the scientist can be divided into three. One is the yeast, second one is the dimorphic fungi, third one is the mold. Yeast best example, Saccharomyces, unicellular, round fungi, reproduction is the budding type, example Saccharomyces. Second thing is the dimorphic fungi. These grow partially yeast, partially filament. The dimorphism, both are there. The meaning of dimorphic, both are present. One is the partially yeast, partially grows as the filamentous fungi. Depending on the condition, environmental condition, climate condition, like this, temperature, pH, like this. These are the under environmental condition. The dimorphic fungi, partially grow yeast like that, partially grow filamentous like that. The best example in this one, Histoplasma capsulatum is the dimorphic fungi. Mold. These are multicellular organisms that are filamentous in nature with hyphae and reproduced through a spores. For best example, Penicillium chrysogenum. Due to their heterotrophic nature, they absorb nutrient from dead material or sometime living matter in a parasitic. Why? All fungi is the heterotrophism. That's where the nutrient from the dead material or living matter in a parasitic or mutualistic relationship with a living organism. Next, fungi create hydrolytic enzymes. Very important. The all fungi to living for the create the hydrophytic or hydrolytic enzymes externally, thus initiating digestion followed by the ingestions. They process a cell wall made up of, already I'll explain the cell wall. The fungal cell wall made up of the chitin and some cell membrane that have stored the steroids. That is the sterol. In the fungi, the type of steroid name is called ergosterol. Sterol is present, but all in the plant sterol, animal sterol like this. But the fungal sterol is called ergosterol, unlike mammalian cell membrane which have cholesterol. See, in the fungal is the which one is the sterols also present. That sterol name is ergosterol. But where the mammalian cells are there, or human beings, animals like that, in that cell, the steroid that is a membrane, the plasma membrane which have the cholesterol is present. The fungi can have very varying modes of reproduction depending on the species type. See, the species type, the fungi, different classification, the mean one is the zygomycotina, mastigomycotina, deuteromycotina, basidiomycotina, ascomycotina, like this. These are the classification of the fungi. Okay, can modes where the you can observe the mode like reproduction. The mode like reproduction depending on the fungi can have varying mode, that is mode of reproduction depending on species type, zygomycotina, basidiomycotina, or zygomycetes, basidiomycetes, and ascomycetes reproduce through sexual reproduction as well as asexual reproduction in some cases also. These three, which one three? Zygomycotina, basidiomycotina, ascomycotina reproduced through sexual reproduction. Only few of them as well as asexual reproduction in only some cases. Through the formation of zygospore, basidiospores, and ascospores. These are the to reproduce the different types of spores. One is the zygospore, second one is the basidiospore, and third one is the ascospores respectively. On the other hand, deuteromycetes are imperfect fungi. Deuteromycetes are imperfect fungi that used to be a subcategory of the Ascomycotina or Ascomycetes. This is a very important, in the Deuteromycotina, here not to direct asexual reproduction like sexual reproduction. Here the name of the Deuteromycotina fungi name, the imperfect fungi that used to be a subcategory, that is the Ascomycetes reproduced asexually through spore, conidia, or by budding. Coming to the next one, multicellular fungi. Multicellular fungi have filamentous structure. The multicellular fungi have filamentous structure and produced by the spores. This is the fungal body. The fungal body name is called mycelium. These filamentous or multicellular fungi mycelium have filamentous structure and produce the spores. The filamentous structures also known as mycelium. Okay, the fungal plant body name is called mycelium. The algal plant body name is called thallus. The fungal plant body name is called mycelium. Are a network of connected hyphae or long chains of cells jointed end to end, divided by a cross walls. After division, these are to form septa. This type of fungi has a vegetative mycelium. Vegetative mycelium which penetrates through the substrate and is responsible for sourcing nutrition. The reproduction mycelium extends outside the substrate into an open atmosphere where it can release the spores. With more than 100,000 known species of fungi, few hundreds are pathogenic to human beings and few thousands are pathogenic to plants. There are more than 1,000 species, 100,000 species are fungi are identified. In that the few hundreds are pathogenic to human beings and few thousands are pathogenic to plants. However, the application of several species of fungi in food and beverages preparation has been carried out centuries. Behind the most commonly known use as mushroom and yeast in food industry. Very important. In food industry, these both fungi are very useful. That is the mushrooms and yeast. Fungi have been used in the creating products like alcoholic beverages, cheese, and bread, etc. What are these are the production like alcoholic beverages, cheese, and bread. This is a general account. What is the traditional food application of fungi? Historical fungi have been consumed for both food and medicinal purpose. In the food industry and pharmaceutical purpose, that is a medicinal purpose with records of various applications of fungi. In ancient Egyptian, Morales and Greek, both scientists, Morales and Greek writings and documentation in Chinese and Joseph traditional medicine. The fungi based fermentation products like soya sauce preparation, tempeh, sake, cozy, onka, Angkor, miso, and humanato have been widely used in Asian cuisines. On the other hand, preparation of mold ripened cheeses like Cambrai, Brie, Roquefort, blue Stillon cheese as well as fermentation of leavened bread using yeast have been common application of the fungi in the Western countries. Instant of fermentation of cereals, that is using for the cereal preparation or beer preparation, alcoholic preparation like these fermentation of cereals using a mixed culture of lactobacillus. This is the bacteria. Mixed culture of the lactobacillus and yeast to produce beers. These are two fermentation. Cereals mixed with the, what are their lactobacillus one and yeast. Both combined to produce to the beer. And fermented porridge can be found in African traditional culture. The production of edible mushrooms, including button mushrooms, shiitake mushrooms, Chinese or straw mushrooms, winter mushrooms, oyster mushrooms, and truffle mushrooms, as well as the production of alcoholic beverages using yeast is a common practice observed across the world. The fermentation as a process can lead to change in a flavor, texture, and other organic properties and improve shelf life and nutrition of food products compared to non-fermented foods. In addition to the production of entirely new products, as in the case of fermentation of carbohydrates leading to the production of alcoholic beverages. While yeast can be particularly attributed to modified flavor, nutritive value, alcoholic content, and living properties, mycelium fungi contribute significantly to external properties, textural properties. The typical food production formed from the fungal fermentation include the beverages, flavораges, protein-rich meat, and substance by a baking production. The fermentation using fungi can be divided into three categories. One is the natural fermentation, starter mediated single stage fermentation, multi-stage fermentation. The first one, natural fermentation, uncooked integrated. Natural is the uncooked ingredients undergo fermentation without the addition of any external fungal culture. Commonly known foods include, idli, dosha, and naan preparation. This is the natural fermentation method. The fermentation occurs due to the presence of microbes in the ingredients or other or the uh, tensile surface. Tensile surface. This is the natural fermentation. Coming to the second one. Started mediated uh, starter mediated. Next one, starter mediated single stage. Starter mediated single stage fermentation method. This one is stage fermentation is the mid second process. These are the, in this case, the ingredient or substrate is cooked and starter media in the form of spores or mycelium is added to it to initiate the fermentation process. The fermented products are cooked before consumption. Tempeh and Orcom are produced by this method. The third one, multi-stage fermentation. In this type of case, solid state fermentation leading to the production of digestive enzymes is followed by liquid state or solid state fermentation involving the breakdown of carbohydrates and proteins. A typical product made from these type of fermentation is rice wine, soya sauce wine, and preparation of the vinegar. This is the multi-stage fermentation. Then the fermentation, one first method is the natural fermentation. That is a commonly example, idli and naan occurs in the presence of microbes, ingredients, tensile surface. Second thing is the starter mediated single fermentation method. This is the cooked and starter media in the form of spores or mycelium is added to it to initiate the fermentation process. These are fermented products are cooked before consumption temp and on proposed by this method. Third one is the multi-stage fermenters. In this case, solid state fermentation leading to the producing the digestive enzymes to preparation from the wine, rice wine, soya sauce, and the vinegar preparation, which above 20% of crude protein content as a percentage of dry matter. The fungi as a food contain all essential amino acids. The presence of the chitin in the cell wall provided a source of dietary fiber. Fungi are also low in fat and lack cholesterol, making them healthy source. See, that's why it used for the low cholesterol and lack of cholesterol, low fat, lack of cholesterol, making them healthy source of the nutrition. What are the overview fungal value chain? And described in the last section, fungi can be used in various food application through a variety of fermentation process. The exciting use case of fungi in the alternative protein sector is that of filamentous fungi for the production of various meat alternative under brands like Quiron, Fermentin, and Promic. Theoretically, yeast can also be used as a protein source for creating alternative protein products. But yeast-based meat alternatives are not in the market yet. Start off like fungi, in ingredients, and more food are exploring yeast-derived protein for creating alternative meat and egg products, while yeast and filamentous fungi have direct application as protein sources. In the alternative protein sector, other fungi-derived ingredients like chitin can also be utilized as sack-flooding materials for the production of cultivated meat, and fungal extract can be used as flavoring and stabilizing agents. The fermentation as a platform can be utilized in the three ways for application of fungi in the alternative protein space. The first route is the traditional fermentation in which fungi are used to ferment a target substrate to improve its sensational textural properties for improved product properties. The second routine is biomass fermentation to produce whole biomass like filamentous fungi and yeast for protein or flavors or textured meat analog. The third route is precision fermentation, which use recombinant DNA technology for exploiting fungi as platforms for production of highly specific compounds. The report focus on alternative protein application of fungi using the first two fermentation routes. The diagram below shows the possible application of the fungi in alternative protein sector and indication to companies working each of the application. See, this is the application of the fungi and all microtechnology and whole cut meat analogs. Next one, bio and protein isolation and flavor components, scarification materials, fermentation processing, and whole out meat analogs. These are the alternative protein application of the fungi. The diagram, it resembles that one. Alternative protein application of fungi. Depending on the application area, the value chain for utilizing of fungi derived ingredients will differ. However, there are three key steps involved in the production of fungi and subsequent utilization of fungi-based ingredient irrespective of the end of application. The process can be broken down into the three broad categories. One is the strain selection involving screening of suitable strains. Production involving biomass production at manufacture scale while maintaining sterile conditions, downstream processing involving destruction of RNA or other toxic compounds, harvesting and concentration of biomass followed by extraction of the target compounds. These have been described this one. The first step, this is the suitable first one, critical step. Critical step for all application areas determines properties of derived ingredients, impact downstream processing. The second one, typically liquid state or semi-solid state fermentation for production of yeast and filamentous fungi. The third one, solid state fermentation for production of fruiting bodies. This next third coming to the last one, the harvesting required for all applications and the RNA removal and sterilization process, extraction of functional ingredients like proteins, enzymes, chitins, etc. for application in meat, alternative and cultivated meat. The specific step involves in the production of yeast and filamentous fungi as a source of protein are similar in nature as depicted in figure 3. Other ingredients like enzymes, chitin can be extracted downstream while the upstream production steps remain the same. The choice of technology at each stage of the value chain will differ due to the difference in the morphological and metabolical properties of the yeast and filamentous fungi. For example, baker's yeast is the typically produced in a fed-bed fermentation system running up to 20 hours while filamentous fungi like Fusarium venenatum is the production in a continuous fermentation system. This running for a period of 6 weeks. Even though both species of fungi can be produced in a liquid state fermentation, the configuration and the type of bioreactors used for many different. See, this is the value of chain of fungi production using liquid state or semi-solid state fermentation. See, these are the semi-solid. This is the RNA reduction. These are the semi-solid and solid state fermentation method. The solid state fermentation are used of the production of mushrooms. See, this is a strain selection. This one is a harvesting, and this is the product development. Product development. Solid state fermentation used for the production of mushrooms have has now found innovative application in packing, furniture, textile, and construction includes several companies like Innovative, Lowell Threading, solid state fermentation. These solid state fermentation through appropriate strain selection followed by optimized of optimized optimized of the fermentation condition and substrate. Companies like Atlas and Photo Teach are creating to the alternative groups, and the next one is the complexity in overall production of process is reduced due to the easy of harvesting biomass. No requirement to concentrate biomass. The animal post process, the mammalian post processing for whole biomass application. That is the, however, the rules of the scaling up or scaling out production system are not as well established as in the case of liquid state fermentation. The value chain for solid state fermentation has been depicted below the figure. This is the the value chain of fungi production, production and downstream operation, downstream, downstream operations are briefly described the following. How to strain? The first one, strain meaning as many now, as with any biological production system, strain selection is the most common systems, most common, uh, crucial step as it determine all the subsequent downstream operations required and impact of the required and required and impact of the required and impact of the processing and genetic productivity required and the growth strain is a lower batch, high growth rate. Key characters of the strain. The first one productivity, the second one genetic stability, third one morphology. The the first one, productivity, high growth rate of strain, as the high growth rate of strain is preferred due to lower batch cycle time and hence faster turn around time for the overall processor. The second item, genetic stability, the ability of the strain to retain its genetic makeup during the complete production cycle is crucial to ensure a stable production process. Third one, morphology. The arrangement is which in which mycelium, the arrangement in which mycelium structure is oxygenized structure is organized impact the mixing process, heat transfer and heat transfer and local nutrient availability. Mycelium can form pellets or filamentous. Depending the process, depending, depending on the process condition, the uh, formation of filamentous leads to higher higher, discovery, hence their power plant requirement to ensure homogeneous mixing of the nutrient to continuous growth biomass. Fourth one is the substrate efficiency. The growth rate of the mycelium. This is the substrate. The growth process. Other important consideration when the selecting appropriate strains for the production of biomass, history of safe use. It is processing. Hmm, it is produced to use strain that is use strain that have a serial history of the safe use and mm, use and are now human comparison. The using a new strain for food applications will require for the significant investment in nutritional and safety trials to establish per regular requirement for the human consumption. A list of species have been historically used for the in the food product has been listed in the table 1. Coming to the presence of mycotoxins. These are the special fungi. Presence of mycotoxins. Fungal species may produce mycotoxins which can cause serious health implications upon consumption. Hence, screening of species that do not produce mycotoxins or identifying a downstream process for selective destructions of mycotoxin would be required to ensure that the fungal biomass is safe for human consumption. In that one, RNA content, it is well known that filamentous fungi and yeast have high RNA content up to 10%. Hence, additional downstream processing steps for reduction or removal of RNA need to be implemented. For food applications, the RNA content in the final product needs to be monitored WHO strands. For maximum allowed RNA content should be complete to it. The strain can be obtained from collection and centers and screening can be performed to identify the most suitable strains. Strains can also be genetically modified or mutated or selectively breed to get desirable trait. The key challenges with genetic engineering fungi for food applications is the concern of the use genetically that is called GMOs. GMOs meaning genetically modified organism for human consumption. In the India context, GMO based food products are not acceptable as per regularly norms in India. What are the production? After the selection of appropriate strain, the choice of the production system will depend on the end application and desired morphology of the end product. The two most commonly used product systems include submerged liquid fermentation and solid state fermentation. What is the submerged liquid fermentation method? And second one is the solid state. Coming to the first one, submerged liquid fermentation is called SLF, S strain submerged, L strain liquid, F meaning fermentation. The fungi grow in an aqueous solution with dissolved nutrients required for the multiplication of the biomass. Microbial fermentation for the production of enzyme is carried out in fermenters or bioreactors without up to 1,000 capacity for application in the alternative protein sector. The intracellular components like proteins of insert in contrast to extracellular components in the fermented both growth as in the case of enzyme production. While the fermentation infrastructure can be utilized for growing fungal biomass, parameters like the concentration of nutrients quality, feedstock, pH, temperature, and oxygen uptake rate need to be optimized to make the process suitable for biomass production and meet safety requirement for subsequent food applications. These parameters can be controlled and monitored continuously during the production process to ensure standard growth condition for all batches. For example, the various types of submerged liquid fermentation available listed in the below. One is the an aerated straight tank fermenters is closed cylindrical tank with mechanical uh, agitation systems like imperial to ensure homogeneous mixing and high mass transfer. High mass transfer rates can be obtained due to the high shear environment created by the aggregation system. High shear may be not desirable of maintaining the morphology of the microorganism is critical as the arrangement of mycelium strands can get destroyed due to the excessive mixing. A bubble column reactor. Bubble column reactor is a vertical erected cylindrical column filled with liquid with a gaseous phase supplied from the bottom of the reactor. Mixing takes place due to the upwards. movement of the gaseous phase through the column. The factor acting in such reactor uh, are lower than in the case of straight tank reactor. Airlift reactors operate on a principle similar to that of bubble column reactor. Mixing in the reactor occur as a result of the circulation of the gaseous phase with a draft tube or in the riser creating gas liquid mixture. The reactor provide a low shear environment and required less fluid in the reactor. The cost of circulating the gaseous phases in the reactor can offset the cost and energy saved due to the lack of agitation system. These reactors do not need internal cooling coils for heat transfer as the external jacket sufficiently sufficiently added by the rapid mixing within the reactor. Next one, fluidized bed reactor consists of a small particles to which cell attach and get immobilized due to the high surface to volume ratio of particles. The oxygen and nutrient transfer rate to the cell are enhanced. These reactor operators at low shear and require low energy for operation. The packed bed reactor consists of a large particles to which cell attached and get immobilized. The particles do not move in the liquid media. This is called the SLF. Can be carried batch, fed batch and another thing. Coming to the solid state fermentation method. This is the second method. As the name suggested, solid state fermentation SSF involving using solid substrate as a growth medium. Due to the use of the solid substrate, fermentation takes place in a low moisture environment, which limitations of mixing and dispersion of nutrient depending on the reactor design. Classical examples of the products produced using solid state fermentation. Induced solid state fermentation include tempeh, mold ripened cheese and mushroom. This type of fermentation is favorable for growing filamentous fungi as these can penetrate solid substrate through the action of hydrolytic enzymes. Hydrolytic enzymes created at the tip of hyphae. Solid state fermentation exhibit high volumetric productivity, low energy consumption, and case of the production downstream processing. However, the disadvantage include limited nutrient, water, and oxygen diffuse, localized heat generation, and limited overall process control that the lead to variable in the final product quality. Through SSF, solid state fermentation, can be carried out in a closed chamber with temperature, humidity control in the place, controlling localized temperature and oxygen moisture availability in challenging. These are the challenging. Despite these limitations, the SSF also due to relatively lower external control required. External control required for SSF. The lower control, low skilled workers can be employed to these lower one can be employed to the SSF cheese like say, next one etc. Hybrid, this one is a traditionally produced household environment within case. Since nutrient is a present, nutrient, water, oxygen, and heat transfer method. The heat transfer and coming to the non-homogeneous SSF, various reactor configurations can be utilized to ensure adequate homogeneity mixing and oxygen supply SSM bioreactor can be classified into four groups. One is the first group, bioreactor without uh, aerations and unforced aerations. Aeration, aerations, aerations. This is the tri-bioreactor. This one is called the tri-bioreactor. The SSF in triobioreactor is carried out in standard, carried out to the carry out to the standard tri-bottom preferred which means to allow the aeration. The thickness of solid substrate on the tray impact heat transfer and gas phase mass transfers and gas phase mass transfer efficiency. The bioreactor without aeration but force aerations. This packed bed reactor consists of a long column filled with solid substrate and perform perforated base at the bottom. Air is forced into the solid substrate from the perforated base, while forced aeration variation variation ensure homogeneous oxygen and moisture is available in the system temperature control and remain a challenge due to the lack of mixing or agitation. The bioreaction with aerations and unforced aeration, rotating drum bioreactor. A rotating drum bioreactor consists of a horizontal, elegant cylindrical drum. This one is okay. This one is the bed reactor. This is the fluidite lead homogeneous mixing. Mhm. These are the bioreactors, fluid bioreactors. Bed reactor consists in the vertical column and plate, which are air is forced upward. The aeration, sufficient aeration is there. The choice of bioreactor used for the production of filamentous fungi using SSF will depend on the extent of homogeneous mixing and temperature, nutrient, water, oxygen control required, while the considering the uh, constraints imposed by the uh, fragility of the mycelium structure formed during the biomass production process. For use of fungal biomass for alternative protein applications, bioreactors with low shear environment for SSF will be preferred. The total process of parameter, biomass quality, growth rate quality, and nutritional value will depend on the parameters duplicated in fungi file, irrespective of the production system used for the production of yeast. The mycelium of the filamentous fungi, the fruiting body filamentous fungi, this is the biological parameters. This is the microbial strain substrate. Second one, physico-chemical parameters, moisture content, temperature, pH, gas phase composition, aeration, and practical particle size. This is a physico-chemical parameters. Next one, mechanical parameter, mixing and agitations. Here, this is the segregation bioreactor content. This is a downstream value chain. Here, harvested cell, spent media here. In between, cell disruptions and selective separations are there. After these two, separation is different, concentration is different. In the separation, cell debris, intracellular products, and to form concentration method. Concentration, one is the extracellular products and purification. This is the total overview downstream value of the chain. Okay. Here also choice for the fermentation techniques influence the complexity downstream process. In this selection downstream operation, relevant for utilization, whole biomass utilization and extraction of intracellular components. Extraction of the intracellular components like mm, proteins and chitin will be discussed with the especially relevant for biomass production, that is called SLF. Keeping keeping in that one, and yeah, useful for the that is the different types. And that one is a cell harvesting. The second one is a cell disruptions. And third one is the pre-extractions. Fourth one is called the selection extraction. Last one, high purity extraction. This one is the different. Cell harvesting method is the different centrifugation, filtrations, sedimentations, and fluorifications. This is the bed milling, homogenations, and the soniocations, autolysis. Next one is the next one enzymatic or chemicals. Chemical or chemically or disruption osmotic potentials. And the coming to the next one, last one is the centrifugation. Centrifugations and filtrations. Last one is the specializations and the chromatography membrane and the chromatography membranes, uh, filtrations, distillations, parations, and the last one, high purity extraction. Chromatography is the high purity extraction, electrophoresis, and dialysis. This total summary of, in this one, the summary of unit operation involved in downstream processing of fungal biomass, adapted in Ball and Franklin scientists. Next, coming to the how to use in these or streams, the harvesting and concentration. After the desired biomass is produced in the SSF biometer, fungal biomass need to be concentrated or further processing. The most commonly used method include centrifugation and filtration. The centrifugations and filtration methods both are two useful and these methods. Two are the both the two uses are there. త్రీ అయ్యిందా? లైవ్ స్ట్రీమ్ స్టాప్ చేయాలి త్రీ అయితే. లీవ్ ద మీటింగ్. త్రీ అయింది కదా.